This is the final video regarding calreticulum expressed on the cell membrane. So far we have understood that calreticulum that are expressed on the cell membrane will cause boost in the immune response in the vitiligo patients, and also helps in the this and also helps in this recruitment of the immune cells, that is CD8 plus T cells and this IFN gamma plus T cells at the site of melanocytes. If you need to understand about this figure, you can refer to our previous video. So today our focus is on whether this calreticulum data expressed on the cell membrane will cause maturation of the dendritic cells or not. And so in order to validate this hypothesis, what they did is they took the samples, or they took the PBMC samples from the patients that are affected with vitiligo and co-culture them in this PIG1 cell line, co-culture them with PIG1 cell lines and introduced this oxidative stress with the help of H2O2. And what they have find out is that they find out different markers for this mature dendritic cells on this PBMC samples that they have used from the vitiligo patients. So first one is basically about this CD8 plus CD80 expressions on the dendritic cells. So in the control case, they can see that with PIG1 and PBMC co-culture without oxidative stress, they can see some signals for the CD80 plus cells, and with the control siRNA, they see control siRNA. It means that the calreticulum is not knocked out, and with the oxidative stress, they see the enhanced CD80 plus cells. And when they use the calreticulum short interfering RNAs that can knock down the calreticulum, they can see the reduction in the signals. They can see the reduction in this CD80 plus signals, indicating that the calreticulum plays a role towards this maturing dendritic cells that will cause the dendritic cells to have CD80 plus and CD11 plus cells. So this analysis is done on the CD11 plus dendritic cells. So in the panel B, they have checked for the CD86, and in the wild type cells they see the 51.8% are CD86 plus cells, and when they use the oxidative stress without calreticulum knockdown, they see the enhancement of the CD80 plus signals, CD86 plus signals, it is up to 75.6%. and when they use the calreticulum knockdown, short interfering RNAs and oxidative stress, they see the reduction is further goes down, so indicating that calreticulum might plays a role towards maturation of the dendritic cells to have the CD80 plus expressions also. And they also check for this HLA-DR. So similar to the other two cases. In the control case, it has 36.0% expression of this HLA-DR plus cells on the CD11 plus dendritic cells, and when they use the oxidative stress, they see the increment up to 55.4%. However, when they knock down the calreticulum, they see the further reduction in this HLA-DR signals. And these things are been quantified, and the difference is statistically significant, that when they use the oxidative stress and without calreticulum knockdown, they see the statistically significant upregulations of the CD80 plus CD11C plus cells and CD86 plus CD11C plus cells, and also HLA-DR plus and CD11C plus cells that can be seen here. So from this experiment, they made a final point that calreticulum are expressed on the cell membrane under oxidative stress, and those calreticulum that are expressed on the cell membrane are responsible for the maturation of the dendritic cells that they have showed using three different populations, such as HLA-DR plus, CD86 plus, CD80 plus, which are the markers for the mature dendritic cells. So this is the final point that they have made in the paper. So I hope you have understood all of these figures, all of the figures that are main figures that are presented in this paper briefly. And if you need to understand more deeply about this paper, how they have conducted their experiments, and what are the supplemental figures represents or depicts, you can refer to the you can go through that paper using the reference that has been provided in the description. So that's all for this video. Thank you for your time.